Okay, so for this discussion, we're going to talk about cellular respiration. Okay, so last time we talked about photosynthesis, the two halves of photosynthesis, which is light reactions, and the second one being Calvin cycle. So now we'll talk about cellular respiration as a whole. So this should be this was supposed to be two modules because it also includes the discussion on the difference between aerobic and anaerobic. But I chose not to focus the discussion on that part, but instead focus on the mechanisms of cellular respiration itself. Okay, so let's start with a simple review of what we discussed for the past few weeks. Let's have first the light reactions. If we can answer each question stated. Okay, first one, what particle is used by plants to supply energy in light reactions? So what particle is used by plants to supply energy in light reactions?
Sir,
Yes po.
photon.
Okay, very good. Light particle or photons. Second, which type of electron flow only involves photosystem 1? Which type of electron flow only involves photosystem 1? Anyone? Anyone? Wala?
Sir,
Yes po.
sir,
Yes.
cyclic electron flow.
Very good. Cyclic electron flow or cyclic flow. Next, what is the name of the special pair of chlorophyll in the reaction center of photosystem 2? What's the name of the special pair of chlorophyll in the reaction center of PS2? Di ba may pangalan yon silang dalawa? Nasa figure.
Sir,
Yes po.
P680 po.
Tama po, P680. Next, oxygen produced in light reactions come from what compound? Oxygen produced in light reactions comes from what compound?
Sir,
Yes po.
water.
Tama po, water. Next, which is the final electron acceptor in light reactions?
Oxygen po.
Sa light reactions, the final electron acceptor. Tama, NADPH. Very good, darling. Okay, so the correct answer is NADPH po. Okay, next. Okay, let's have the Calvin cycle. Okay, number one, how many ATPs are spent in the reduction phase considering one molecule of carbon dioxide? How many ATPs are spent in the reduction phase considering one molecule of carbon dioxide?
Sir,
Yes,
2.
2 po. That's correct. Second, how many is the net gain of G3P for every 3 molecules of CO2 entering the Calvin cycle? How many is the net gain of G3P for every 3 molecules of CO2 entering the Calvin cycle?
Sir, 1.
Very good, 1. Third, which enzyme catalyzes the reaction between RUBP or ribulose bisphosphate and carbon dioxide?
Sir,
Yes po.
Robisco po.
Very good, Rubisco. Fourth, in total, how many carbon dioxide are needed to enter the Calvin cycle? to create a simple sugar like glucose. In total, how
Sir,
many? Yes po.
6 po.
6. Very good. Last, what is the role of ATP in the reduction and regeneration phase? What is the role of ATP in the reduction and regeneration phase? Ano ba ginagawa ng ATP dun sa mga compounds sa Calvin cycle?
Sir,
Yes po.
it provides energy and phosphate ion.
Okay, so nagbibigay siya ng phosphate or it phosphorylates yung compound. It phosphorylates the compound. Okay, so very good. So medyo fresh pa naman kahit papaano sa utak nyo yung two topics natin. Okay, so very similar ang cellular respiration pagdating dito sa mga cycles and especially dun sa electron transport chain. Pero kasi sa photosynthesis, hindi natin masyado na-discuss yung pinaka um, mechanism niya. Dito, mas titignan natin siya in detail. Okay? So, doon medyo described lang. Dito titignan natin more on detail. Okay? So, cellular respiration, the term cellular respiration itself, technically, um, it involves both aerobic and anaerobic respiration. Okay? But dahil yung cellular respiration minsan nakikita siya ng most na tao as yun nga 'di ba parang paghinga or similar to yung pag-respire talaga natin via lungs. Usually it is the cellular respiration itself is mostly associated with aerobic respiration. But in technical terms or in technicalities, it also involves anaerobic respiration. Okay, so simply the difference between the two is that one can happen without the presence of oxygen or with low presence of oxygen. That's for anaerobic, and the aerobic involves the presence of oxygen. Okay? So for anaerobic, sabi dito, it also involves the partial degradation of sugar and it includes processes like fermentation. Okay, for aerobic, an organic fuel is consumed. So this along with oxygen. So they are both reactants and they are both consumed in the reaction. Okay, so mostly glucose or sugar yung kinokonsume din dito. Okay, so this is the general equation for cellular respiration. Okay, glucose plus 6 moles of oxygen will yield 6 moles of carbon dioxide, 6 moles of water, energy in the form of ATP, as well as, of course, heat, because the second law of thermodynamics. Okay, so even in the figure on the right, as you can see, um, hindi parang makikita din natin dito that for every system talaga, energy conversion is somehow inefficient because of the second law of thermodynamics. But considering yung mga man-made machines natin and such, cellular respiration itself or aerobic respiration is one of the most efficient types of energy conversions there is on Earth. Okay, so kahit na siya I think it's efficiency is around 32%. So kahit na nandun lang yung efficiency nung pag-convert niya ng energy, it is one of the highest energy conversions na nandito na nakikita sa planet natin. Okay? So yan, on the figure on the right, as you can see, light energy enters the system and ito yung energy from light is somehow kumbaga embedded sa mga cells or tissues natin via photosynthesis kasi dito na nata-translate into an organic substance or compound yung energy na yon. Okay? And then pag meron ka na nun, we can use that as a source of energy. But that conversion again is um, not 100%. Okay? It is one of the most efficient but hindi siya 100%. So some energy will still be lost as heat. So this is the general overview for cellular respiration. Technically, we have three parts. Parang in most cases or in most discussions, um, nadi-divide talaga siya into three parts. Yung glycolysis, yung Krebs cycle or citric acid cycle, tas yung ETC. But in general, or actually, yung tama is hindi talaga ETC yung third phase niya, kundi yung, yan, yung tinatawag na oxidative phosphorylation. Ito yung pag-produce talaga natin ng mas madami. At ito talaga yung pag-produce natin ng maraming ATP. Okay, so ito yung may highest na yield ng ATP sa buong um, sa kabuuan ng cellular respiration. Kasi yung glycolysis and yung citric acid cycle o yung Krebs cycle, parehas nakakapag-produce na ATP yan, pero medyo onte as compared to oxidative phosphorylation. Tapos yung ETC kasi, part lang siya ng oxidative phosphorylation. Yung ETC itself is just a mechanism to ensure yung tinatawag na chemiosmosis. So, para mangyari yung chemiosmosis, yung ETC yung mechanism na nag nagpo-provide ng mga conditions para mangyari yung chemiosmosis. So, pag pinagsama mo yung dalawa, yun yung nakakapag-produce ng maraming ATP. So, yung third phase should be oxidative phosphorylation. But there's a short transition stage or transition phase between glycolysis and Krebs cycle, and that is yung tinatawag na pyruvate oxidation, kung nakikita nyo. So, meron pang transition stage, yung pyruvate oxidation, to which yung pyruvate, ma-oxidize nga siya to form acetyl coenzyme A. Okay? So, let's just differentiate the two. Itong substrate level and oxidative phosphorylation. So, like I said, sa glycolysis and sa Krebs cycle, parehas nakakapag-produce ng ATP. Pero, ang pinagkaiba nila is dito sa dalawang to, substrate level lang as opposed dito sa third phase, oxidative phosphorylation siya. So what does that mean? Pag substrate level phosphorylation, it involves an enzyme, okay? Kaya nga siya substrate level. So may involvement ng enzyme yung pag-create ng ATP o yung pag-phosphorylate ng ADP, yung pagkabit ng isang inorganic phosphate sa ADP, enzyme yung involved. And most of them, tawag doon ay kinase. Kapag siya ay nagpo-phosphorylate, usually that group of enzyme is called a kinase. So, pag may kinase sa dulo, yung pangalan, usually, nagpo-phosphorylate yung enzyme na yun. So, dito sa glycolysis and sa Krebs cycle, parehas may enzyme na nakapag-produce ng ATP via substrate level phosphorylation. Pero pagdating sa third phase, kasi nga oxidative phosphorylation siya, merong redox reactions na involved, meaning may pagpasa-pasa or may pag-accept ng electron. At the same time, yung final electron acceptor dito sa oxidative phosphorylation ay oxygen. Okay? Kaya isa sa mga byproducts ng cellular respiration ay water. Kasi once oxygen accepts the electron, nagiging water siya. Okay? So, most probably because the electron is in the form of a hydrogen ion. Okay? So, yon. Pag oxidative phosphorylation involve ay redox reactions. Kapag naman substrate level phosphorylation involve ay enzymes called kinase. Okay? Yan. So, kung titignan nyo, similar to light reactions, yung products ng glycolysis and ng Krebs cycle or ng citric acid cycle, ito yon, NADH and FADH2. Sila yung parehas na nagsosupply doon ng electron at ng hydrogen ions dun sa oxidative phosphorylation na stage. Okay? So, similar to light reactions kasi si light reactions, siya yung nagpo-produce ng NADPH and ATP for na Calvin cycle. So dito naman, NADH and FADH2 yung ginagamit for oxidative phosphorylation. Okay, so let's look at glycolysis. Sa glycolysis, again, substrate level phosphorylation siya. Okay, so nakapag-produce tayo ng ATP using an enzyme. And glycolysis is divided into two phases. Okay, so it's divided into two phases, yung tinatawag na energy investment phase and energy payoff phase. Okay, so sa energy investment phase, basically, may investment ka ng dalawang ATP. So somewhere between yung conversions ng glucose to different compounds, kailangan gumamit ng dalawang ATP. Makikita natin mamaya kung anong process yung kinailangan ng energy dyan. So, may investment tayo na 2 ATP. Pero down the line, pagdating sa energy payoff phase, makakapag-produce tayo naman ng 4 ATP. Therefore, nabawi natin yung ininvest natin na dalawa. So, kumita lang din tayo ng dalawang ATP. At the same time, dito sa payoff phase, makakapag-produce ka ng dalawang NADH. Okay, 2 NADH. But the final product for glycolysis would be pyruvic acid or pyruvate. And from one molecule of glucose, makakapag-produce ka ng dalawang pyruvate. Okay, so from one molecule of glucose, may dalawa kang pyruvate. Plus 2 water molecules. Okay, so ito yung net gain natin. Nakikita nyo sa baba, the net gain would be for one molecule of glucose, you produce 2 pyruvate, 2 water molecules. Tapos, ang net gain natin sa ATP ay dalawa lang din. Tapos, ang gain natin sa NADH ay dalawa din. Okay? So, bakit naging dalawa lang yung gain sa ATP? Because nag-invest tayo ng dalawa dun sa energy investment phase. So, 4 minus 2 equals 2. Okay? So, ayan, nakalagay dito, involves various enzymes which phosphorylates. So, may kinases dito, isomerizes. So, isomerases, may mga nagki-cleaves o yung mga aldolase ata to, and such. Okay, so tignan natin. So, in reality, ito talaga yung nangyari dyan. Madaming enzymes and madaming compounds na involved. So, kung makikita nyo, dito tayo gumamit ng unang ATP natin when converting glucose to glucose 6-phosphate or basically nag-add lang tayo ng phosphate group doon sa glucose. Kaya siya naging glucose 6-phosphate. Kaya gumamit ng ATP. Okay, so kung makikita nyo, hexokinase. So, kinase yung enzyme na involved. Okay, nagdagdag siya ng phosphate group dun sa glucose. Okay, so sumunod na enzyme, may isomerase. Okay, third, pagdating dito, kinailangan ulit ng ATP when converting fructose 6-phosphate to fructose 1,6-bisphosphate. Okay, so dalawang phosphate group na yung meron siya. Nag-add ulit tayo ng phosphate through phosphofructokinase, another kinase enzyme which phosphorylated fructose 6-phosphate, forming fructose 1,6-bisphosphate. Okay, so ito yung fourth enzyme, aldolase, cleave niya daw yung fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate or DHAP. Okay? Pero itong DHAP kasi, nako-convert din siya into G3P. So, kumbaga kung iisipin din natin, parehas ding magiging G3P yung product dito. Kasi itong DHAP, i-convert din siya back into G3P. So, yun yung mag-move sa mga next steps. Okay? So, pagdating dito sa triose phosphate dehydrogenase, doon nakapag-form ng dalawang NADH. Okay? So, kung makikita nyo, sa next na mga steps, nakapag-produce ka na ng dalawang ATP dito sa phosphoglycerokinase. And then dun sa last one, yung pyruvate kinase, nakapag-produce ka din ng dalawang ATP. And the final product for glycolysis would be your pyruvate. Okay? Okay, so kung yung final product sa glycolysis ay pyruvate, yung first na reactant pagdating sa Krebs cycle or sa citric acid cycle ay obviously ay pyruvate. But before it enters the cycle itself, dadaan muna siya sa isang short transition stage or transition phase, which is yung oxidation ng pyruvate to acetyl coenzyme A. And pagdating dito, kung mapapansin nyo, merong pag-produce pa rin ng NADH. Okay, so yung oxidation ng pyruvate, meron ka pa ring mapaproduce na NADH. So what from glycolysis, meron ka ng dalawang pyruvate na papasok diyan. Okay? So isang pyruvate magiging acetyl coenzyme A, so isang pyruvate magiging acetyl coenzyme A. So sa transition stage, meron ka na kagad dalawang NADH. Okay, so sa transition stage to dalawang NADH kagad. So assume natin na dalawa na kagad siya diyan. Okay, so dito bago siya pumasok sa Krebs cycle, magiging acetyl coenzyme A muna siya. Okay, so ang papasok mismo sa cycle ay CoA na, acetyl CoA. And if you notice dito, tignan muna natin yung mga products dito sa Krebs cycle. Per conversions na nangyayari, nakakapag-produce sila ng NADH, FADH2. Okay, so yan yung mga magsusupply ng electrons dun sa electron transport chain natin. And then eventually, makapag-produce ng gradient para sa chemiosmosis natin. Okay? Okay. Ayan. So ito talaga yung pinaka tawag dito. Pinaka conversions or yung mga processes na nangyayari pagdating sa Krebs cycle or sa citric acid cycle. Okay, so yung acetyl coenzyme A, basically pag enter niya sa cycle, kino-convert siya to citrate. Yun yung first product, kaya siya naging citric acid cycle. Pero sometimes it is also called as tricarboxylic acid cycle. Okay, sometimes yan yung tawag din sa kanya because yung citrate ay or yung citric acid ay isang tricarboxylic acid. So, pwede din siyang tawaging tricarboxylic acid cycle. So, citric acid cycle, Krebs cycle, or tricarboxylic acid cycle, they're all the same cycles. Okay? So, kung makikita nyo, basically, yung citrate natin, i-convert lang natin siya to different compounds. So, kino-convert lang siya to different compounds, citrate to isocitrate, to alpha ketoglutarate, succinyl CoA, succinate, fumarate, malate, oxaloacetate, sorry. So basically, kino-convert lang siya. So kaya nagsa-cycle lang siya. But doon sa conversions ng compound via the citric acid cycle, the Krebs cycle, nakakapag-produce sila ng NADH o ng mga electron carriers. Okay, so NADH, FADH2, lahat yan ay yung byproducts na napoproduce ng citric acid cycle na gagamitin natin for electron transport chain and for oxidative phosphorylation. Okay? Sa glycolysis, NADH lang yung na-form. Dito sa citric acid cycle, NADH and FADH2. Okay? So, parehas electron carrier na gagamitin for oxidative phosphorylation. And dito sa Krebs cycle, meron din substrate level phosphorylation. So, ibig sabihin nakapag-produce tayo ng ATP. Okay, so saang stage dito po? Sa conversion ng succinyl CoA to succinate. So, meron tayong napoproduce dyan na ATP. Okay, so kung makikita nyo, meron pa dyan GTP, GDP, ganyan. So, yung GTP, para din siyang ATP actually, but here, it is converted into ATP. Okay, so yung GTP convert siya to ATP. Okay, so yun yung byproducts natin for citric acid cycle or Krebs cycle. Okay, now let's move to the electron transport chain, and chemiosmosis or oxidative phosphorylation. Okay, so yung mga products natin sa citric acid cycle and glycolysis, which are your electron carriers, namely yung NADH and FADH2, sila yung gagamitin nating power supply para sa electron transport chain natin, and then eventually our chemiosmosis. Okay? So, doon sa mitochondria. Nakalimutan pala natin sabihin ko sa nangyayari yung mga yon. Okay, sorry. Dito, tignan natin. Ayan, dito. So, glycolysis actually happens outside of the mitochondria. Okay, glycolysis outside of the mitochondria. Yung pyruvate oxidation, Krebs cycle, and oxidative phosphorylation, they all happen inside the mitochondria. Si oxidative phosphorylation o yung ETC natin mismo, siya ay nahahanap sa inner membrane. Okay, inner membrane ng mitochondria. Tandaan, inner membrane ng mitochondria yung electron transport chain. Ibig sabihin, yung mga proteins natin doon sa electron transport chain, yung mga protein complexes natin, naka-embed siya doon sa inner membrane ng mitochondria. Kung sa light reactions, siya ay nasa thylakoid. Dito naman sa cellular respiration, siya ay nasa inner membrane ng mitochondria. Okay? Okay, so balik tayo ulit. Dito sa ETC, yung mga protein complexes natin, may mga kanya-kanyang pangalan yan. Pero pwede naman siyang tawagin as protein complex 1, 2, 3, and 4. Okay? So, itong mga to, yung protein complexes natin, may kanya-kanya silang electronegativity. Meaning, yung mga protein complexes na to, may affinity sila to electrons. So, sinasabi that protein complex 1 is the least electronegative, tapos protein complex 4 would be the most electronegative. Okay, so pababa yung movement niya. Pero yung last electron acceptor natin, which is oxygen, siya yung considered talaga as most electronegative, meaning mataas yung affinity niya to electron. Pag present si electron, accept talaga niya ng buong puso. Pero pagdating kasi dito, kaya lang gusto ko ipakita tong electronegativity niya. Because yung pagpasa-pasa ng electron from one protein complex to another, napapower siya through this relationship din kasi. That each protein complex is more electronegative than the other. So, pag kailangan niya na ipasa, willingly i-accept ng susunod, and then i-accept ulit ng susunod, i-accept ulit ng susunod. Okay, so parang ganun lang yung idea natin. So, from our electron carriers, like NADH and FADH2, pag pinasa na nila yung electron to the next protein complex, willingly na-accept nung next na protein complex because they are more electronegative than the previous one. Okay? So, eto mga to, kung makikita nyo, yung Q, yung CYT o yung cytochrome, yung mga yan ay mga tinatawag na prosthetic groups. So, kung naalala nyo yung discussion sa enzyme, yung mga prosthetic groups, pwede silang organic or inorganic na molecules na ginagamit ng enzymes para mag-function ng maayos. Okay, so itong mga protein complexes na to, sorry, not enzymes, proteins. So yung mga prosthetic groups na to, ginagamit siya ng mga proteins para efficiently silang mag-function. So itong mga nandito, yan ay mga prosthetic groups na tumutulong mag-function para mag-function ng maayos yung mga protein complexes dito. Okay? So, kada pasa ng electron for each protein complex, yung ating protein complex, nagre-release yan ng hydrogen ion towards the intermembrane space. Ito, dito sa space na to. 'Di ba, ito inner membrane to ng mitochondria. Ito intermembrane space. Yung nasa dulo niyan, yun yung outer membrane. So basically, yung intermembrane space ay yung space between sa inner and outer membrane ng mitochondria. Kaya nga siya tinawag na intermembrane space. Okay? So each accept ng electron ng protein complex natin, nakakapag-pump sila ng hydrogen ions towards the intermembrane space. Okay? So dito sa intermembrane space, aasahan mo madaming hydrogen ions or mataas yung concentration ng hydrogen ions sa intermembrane space natin. Okay? Because of the protein complexes in our electron transport chain. Wait lang. Okay. So dito, so kung dumadami yung mga yung mga protons natin o yung hydrogen ions natin sa intermembrane space, nakaka-create sila ng gradient. Okay? Bakit sir nagkakaroon ng gradient? Because mataas yung concentration dito sa intermembrane space, pero pagdating dito sa loob ng mitochondria, mababa yung concentration ng hydrogen ions. So may difference. Dito mataas, dito mababa. So nagkakaroon ng differences sa concentration nila. That's why nagkakaroon ng tinatawag natin na gradient. Okay? And through this gradient, nakaka-create ng natural na force yung mitochondria natin. And tawag natin doon sa force na yon ay proton motive force. Okay? Proton motive force. Si proton motive force, kaya ko siya binanggit because basically siya yung natural na force na ginagamit ng mitochondria to undergo chemio-osmosis. Okay, so sige, i-clear muna natin saglit. So si proton motive force, 'di ba, mataas yung concentration ng protons dito o ng hydrogen ions natin. Tandaan, positively charged siya. Okay, question ngayon. If yung mga ions dito puro positive, ano yung magiging reaction nila towards each other? Okay, let's say medyo maglapit itong dalawang hydrogen ions na to. What would be their reaction towards each other? These two hydrogen ions. What do you think would happen if magkalapit sila?
Sir,
Yes po.
magiging hydrogen gas?
Let's look at it this way. Paano ba? They are both positive in charge. Sige, yun yung tignan nyo. They are both positive in charge. So, once na sila ay nagtabi or nagkalapit, what do you think would happen to both of them? They are both positively charged. So, like sige, sabihin natin kunyari sa magnet, ganyan. If they have the same, ano ba yun? Polarity ba? What do you think would happen? Since they have the same charge, they would repel. Very good. They would repel or yung kumbaga, yun nga, repel or kumbaga pang ano sila, they would move away from each other. Okay? Not towards. Hindi sila mag-attract towards each other because they have the same charge. So they would repel. So dito sa intermembrane space, since mataas yung concentration ng hydrogen ions o ng protons natin, basically, medyo malikot sila dyan because they would be repelling against each other. Medyo malikot sila dyan kasi ang gusto talaga nilang mangyari, mag-move towards dito inside. Okay? Ayaw nilang ma-stuck dito sa intermembrane space. Pero, hindi kasi sila readily nagpa-pass through dito sa phospholipid bilayer. Dito sa phospholipid bilayer na to, hindi sila pwede mag-pass through dyan. Kaya, the only way that they could pass through is through this complex protein called, or complex enzyme called ATP synthase. Okay? ATP synthase. So this is an enzyme na medyo complicated yung machinery niya. Ito lang yung way para makapasok yung hydrogen ion natin papunta sa loob ulit ng mitochondria. Okay? And once na pumasok yung isang hydrogen ion sa ATP synthase natin, nagkakaroon ng mga tinatawag na conformational changes yung enzyme or nagbabago siya ng shape, nagbabago ng anyo. And yung pagbabago ng anyo niya na yon, actually, may kaakibat na movement yon or may kaakibat siya na, yun nga, na actual motion nung enzyme. And through this motion or through that movement or through that work, nakakapag-attach yung nakakapag-attach yung enzyme ng isang phosphate group sa ADP, therefore producing ATP. So, yung movement ng ions o yung movement ng protons, ng hydrogen ions from the intermembrane space papaloob through ATP synthase, yun yung mechanism to which nakakapag-attach ng phosphate group to ADP. Okay? So, hindi ka gumamit ng energy. Ang ginamit mo lang is yung proton motive force. Yung movement ng mga protons o ng hydrogen ions doon sa intermembrane space, yun yung ni-utilize ni mitochondria para makapag-produce ng ATP. So, wala siyang ginamit na energy but instead yung natural na gradient force o yung proton motive force yung ginamit niya. Okay? Paano niya nagawa yon? Gumawa siya ng tinatawag na ATP synthase o yung enzyme na nakakapag attach ng phosphate group to ADP by simply moving or by simply conforming um, every time na may pumapasok na hydrogen ions sa kanya. Okay? So every time na may pumasok na hydrogen ions diyan, may movement na nangyayari na nag-e-enable sa kanyang mag-attach ng phosphate group to ADP. Okay? So ayan yung description niya nasa gilid. You can read that. But hindi ko na siya di-discuss ng buo. Basta alam natin that yung movement ng hydrogen ions papaloob via ATP synthase nakakapag-produce ng ATP. So, bawat hydrogen ion or proton na inaambag nitong mga protein complexes, 'di ba? Lahat 'yan nagpa-pump ng hydrogen ion sa intermembrane space. So, yung mga 'yan, basically, itong mga hydrogen ions ngayon yung ginagamit ng ATP synthase para makapag-attach ng phosphate group to ADP, forming ATP. Okay? That's why yung mga byproducts ng Calvin cycle, ng Krebs cycle at ng glycolysis, namely yung NADH at FADH2, yun yung nagsusupply ngayon ng energy or yun yung nagsusupply ng way para makapag-produce tayo ng ATP. Sila yung nagsusupply ng proton or ng hydrogen ions to produce ATP. Okay? So, tignan natin. Ayan, dito mas maayos lang yung figure. So, gaya ng sabi ko sa inyo, itong mga electron carriers na to, magpapasa-pasa yung electron from one protein complex to another with varying electronegativities. Okay? Ito pala, ganyan pala siya. Yan. So, itong mga to, mga hydrogen ions or protons, sila yung masastock ngayon dito sa intermembrane space. Okay? Ito, outer membrane to. So, itong movement or random movement ng mga protons natin na gustong pumunta sa loob um, ng mitochondria, yun yung nagsusupply ng way para makapag-attach tayo ng phosphate group to ADP producing ATP. So, yun yung chemiosmosis na part. Okay, yun yung chemiosmosis na part. Yung pag-move ng protons o ng hydrogen ions papaloob ng mitochondria via the ATP synthase. And through that movement, nakakapag-produce tayo ng ATP. Okay? Yan o. ATP synthesis powered by the flow of hydrogen ions or protons back across the membrane o yung papaloob. Okay? If you notice, NADH, it directly donates its electron to complex 1, protein complex 1. Pero si FADH2, sa complex 2 siya kagad. Hindi siya dumadaan sa complex 1. Okay? So, I guess that's all for the electron transport chain, and chemiosmosis, or collectively oxidative phosphorylation. Okay? Ay, ito pala, sorry. Forgot to mention, yung final na electron acceptor dito sa electron transport chain ay oxygen, forming water. So, dito na form yung water na byproduct ng cellular respiration. Okay, let's account the ATP. When accounting for ATP, mananotice natin that for one molecule of glucose, mananotice natin that for one molecule of glucose, hindi sure yung number. Pwedeng 26, 28 pagdating sa oxidative phosphorylation. Tapos kapag sinam mo na siya, pwedeng siya ay 30 or 32 ATP. Okay? For one molecule of glucose. So, ito daw yung mga reasons kung bakit nagva-vary yung number ng ATP. Okay? So, ito yung mga sabi ng mga scientists natin. The first reason is that the ratio of NADH to the number of ATP is not exact. Meaning, yung nabibigay daw na ATP per electron carrier, kasi itong mga NADH na to, 'di ba? May kaakibat na napoproduce yan na proton. And sabi, hindi daw exact yung naaambag na hindi sure kung ano yung exact na value ng naaambag na proton per molecule of NADH. So, pwedeng, nung una sabi daw around, I think, 8 NADH, ay 8 protons daw yon. Pero parang nagkaroon ng bagong change na sinabi na ang agreed na daw is around 4 protons yung naaambag niya. Tapos, for each number of proton, hindi din exact yung number, For each number of proton, hindi din exact yung number of ATP na napoproduce niya. I think ang ganyan yung number niya, 2.5 ATP. So for each proton na dadaan sa ATP synthase, around that number daw yung napoproduce na ATP. So hindi din exact talaga. The next reason is that yung yield daw nagva-vary depending sa type of shuttle used to transport electrons. So, yun nga, NADH, nagva-vary yung kanyang inaambag na protons, therefore nagva-vary din yung ATP na napoproduce niya. As well as yung FADH. So, depende dun daw sa shuttle na ginagamit. Okay? So, hindi, iba pala to, sorry. Yung NADH, hindi daw siya readily na papasok dito sa mitochondria. So, yung NADH dito sa glycolysis, kailangan siyang i-shuttle papaloob. So, depende daw sa magta-transport sa kanya. Kung NAD or FAD. Okay? So, yung electron mula dito sa NADH sa glycolysis, ipapasa to a shuttle, either NAD or FAD. So, depende daw dyan, kung ano yung magsha-shuttle dyan, yung magiging yield ng ATP. Kasi per cell or per type ng tissue, for example, brain tissue or liver or heart tissue, magkakaiba sila ng shuttle na ginagamit. Magkakaiba. So, magkakaiba din ng yield. Depende kasi dun sa tissue or sa cell kung kailangan niya ba ng maraming energy or kailangan niya ng sakto lang na energy. So, kung matapos siya sa energy, ibang shuttle yung gagamitin niya. I think NAD yung gagamitin niya. Next, so, the third reason would be yung proton motive force na napoproduce natin sa oxidative phosphorylation for chemiosmosis, hindi siya lahat ginagamit for producing ATP. Yung proton motive force na napoproduce ng ETC natin, hindi siya ginagamit for chemiosmosis lahat or 100%. sometimes ginagamit siya for uptake or other processes. Ano yung uptake, sir? Uptake, for example, kapag yung pyruvate, mag-enter na siya ng mitochondria or the uptake of pyruvate, gagamit tayo ng energy dyan. And yung energy na ginagamit dyan, pwedeng ang mag-supply nun ay proton motive force. So kung 100% ng proton motive force ay iaalay lang natin for ATP production or for chemiosmosis, most probably daw, I think around 28 yung mapoproduce natin. Pero kung hindi, around 26 lang daw. Pero yun nga, may iba-ibang reason pa kung bakit pwedeng mag-26 siya or mag-28 siya, which is this and this one. So, kaya nagva-vary. Pero I think sa mga modules or dun, sa module nyo, I think 32 na yung sinasabi. But yun nga, pwedeng mag-vary talaga siya. Either 30 or 32. Okay, so mas tama to, 30 or 32. Okay? So dito, babanggitin ko lang yung sa anaerobic respiration kasi hindi talaga ako masyado nag-focus dito. For anaerobic respiration o yung absence ng oxygen, ito yung mga tinatawag nating mga fermentation processes. And by two types siya, alcohol fermentation and lactic acid fermentation. Sorry, lactic acid fermentation. For alcohol fermentation, obviously, ang byproduct nito ay ethanol or alcohol itself. For lactic acid naman, lactate or lactic acid itself. Okay? So, kung makikita nyo, yung glycolysis, anaerobic talaga yan in nature. Kahit sa aerobic respiration na part, yung glycolysis ay anaerobic kasi wala pa namang oxygen na involved doon. Pero dito sa anaerobic respiration talaga, nakapag-produce tayo ng NADH kahit kumbaga kahit medyo incomplete yung pag-process natin dun sa glucose. So kahit medyo incomplete siya, nakapag-produce pa rin ng NADH dito sa anaerobic respiration. Okay? So, yung alcohol fermentation, yan yung ni-utilize usually sa industries, like yung mga beer making, wine making, and such. Etong lactic acid, ang example dyan is yung build-up ng lactic acid sa muscles nyo. Yung nagkakos ng soreness ng muscle or ngawit, lactic acid build-up yon sa mga muscles. So, basically kasi, pag nag-exercise tayo or tayo ay nagwo-workout, yung aerobic respiration, sometimes hindi siya enough to supply energy for our muscles. So, nauubos na yung oxygen sa tissues natin kasi mas nagagamit siya. Kumbaga, yung pasok ng oxygen ay kulang. Yung supply ng oxygen ay kulang compare sa demand niya. That's why yung tissues natin nag-undergo siya ng anaerobic respiration, to be specific, ng lactic acid na process or lactic acid fermentation. Kasi nga kulang sa energy. So kailangan niyang mag-produce pa ng energy kahit walang oxygen kasi nagwo-workout tayo. So yon, yung soreness na naramdaman or yung ngawit na naramdaman sa muscles, that is because of lactic acid buildup. Okay? Okay, so let me just stop my share for a while. I may stop recording.